All right, to kick off the afternoon session, we have Jiyoung Lee from the Division of Environmental Health Sciences, College of Public Health and Department of Food Science and Technology, College of Food, Agricultural, and Environmental Sciences, The Ohio State University, about the role of wetlands in mitigating pathogens, a new topic for us today. So thank you very much.
Thank you. So it's kind of hard to see my slides when I stand up on the podium, so let me talk from here. Thank you for this opportunity, and then see the audience. Okay, so the outline of today's talk, I'm going to talk about more health-related thing. So why do we care? Harmful algal bloom and those toxins. So a little bit of introduction, and then I will go with specifically how it is related to wetlands. Can wetlands do something with harmful algal bloom reduction? We learned about all the nutrient reduction, then actually harmful algal bloom, it can be some mitigated. But wetlands, before that, I'm going to talk a little bit about enteric pathogens, but indicator E. coli and their nutrient pathogens and harmful algal bloom. So the audience, I think most of us, we know about it, but freshwater harmful algal bloom is different from marine one. But in freshwater, it is the freshwater harmful algal bloom we do care because freshwater, that source is used for our agriculture, drinking water, recreational, and tourism, and a lot of things. So this is actually beyond Ohio, beyond US. It is a global problem. And the reason why we do care for now more is that the intensity, frequency, prevalence, all those things are increasing. So I'm originally from South Korea. So South Korea, China, and Australia, or Guatemala, and here, all we face the similar problem. So in freshwater, who are the major players? Actually, we call them harmful algae bloom. But algae are eukaryotes, but those we see more chronically in Ohio and other countries are cyanobacteria. So these are the major players who make the bloom toxic. And then I'd like to focus on Mycocystis because that's the most prevalent one in Ohio and also other parts of the world. But in Ohio, Mycocystis is the #1 player, and second is Plankthorax. So Mycocystis is in Western Lake Erie. We see them more often, but Plankthorax in inland lakes such as Grand Lake St. Mary and Buckeye Lake. So cyanotoxins are the reason why we do care in terms of human health, animal health, even plant health impact. So among the cyanotoxins, depending on how they affect, so there are a couple kind of types. One is hepatotoxin, that means it gives toxic effects to liver. So mycocystin is one of them. And then other cytotoxin, neurotoxin, and dermatotoxin. So mycocystin is the one a lot of, very much prevalent. There are many reasons. Their chemical structure is very stable, so they can be more persistent in environments. And also, a lot of the most predominant cyanobacteria, they make mycotoxins. So there are many reasons why I do care more about mycocystin. So among the mycocystin, depending on what kind of congeners, they show different toxicity. The most common thing we hear about is MCLR. We also hear about MCRR. Depending on their congeners, their toxicity is different a little bit. But microcystin LR is the most kind of common, but also most studied. So adverse effects, it can cause chronic and acute effects. One of the chronic effects is if we expose at the high level of microcystin for length of time, it can develop liver cancer. But acute symptoms are more like a liver failure. And then if acutely exposed to, it causes a lot of like rashes and then skin and eye, stomach upset. So a lot of symptoms, acute symptoms, can be really confused with other causes. And then also microcystin also affects our endocrine system, so it can cause reproductive related cancer or other disease. So first, we need to monitor. Dr. Jason Marion is in the audience. His initial work was about developing rapidly monitor harmful algal bloom in fresh water. So one of his doctoral dissertation was phycocyanin. He developed a model based on phycocyanin and then other factors. So phycocyanin measurements can be done within just a couple of minutes. So by detecting phycocyanin, we can predict elevated level of microcystin in inland lakes. So we prove that. So using that kind of phycocyanin also, but what if we need to monitor a lot of lakes throughout the state, throughout the nation, or what if we need multi-year data? So we kind of moved to cellular remote sensing, focusing either phycocyanin or even chlorophyll. And then using that one, Feng Zhang was one of my doctoral students. One of her dissertation was using cellular remote sensing, then can we do drinking water intake? So we did some kind of validation study with a lab measurements, looking at can we do kind of elevated level of toxin tracking using cellular remote sensing. And then we linked to that kind of data across the U.S. Freshwater lakes, and then liver disease-related motility. So then we found that 63% of U.S. counties showed detectable level of bloom coverage. Then what if non-alcoholic liver disease? And then we found significant clusters, about 65 spatial clusters. And then we saw that the counties in the clusters also show higher level of bloom coverage. Then we kind of developed some model and then looking at whether there's a linkage of the bloom coverage and non-alcoholic liver disease motility. And then you can see that the kind of the red dots in Lake Okeechobee area and the Lake Erie. And then there was a positive and significant correlation. And then if the county has 1% increase of bloom coverage, we saw that 0.3% increase of non-alcoholic liver disease motility after adjusting all confounding factors. So we have like a rationale why we need to care about harmful algal bloom. We need to mitigate it. And then also the morning session talking about the root cause is nutrient eutrophication, so wetland can do it. So the wetlands definitely can provide ecosystem service, but can wetlands provide public health service? So in terms of ecosystem service, so they can, we know that a lot of data accumulate that. It can provide ecosystem service. We are talking about nutrient reduction. Yes, we have all the evidence. Go back there. So one of the, another service is it attracts a lot of wildlife, including all the migratory birds and other animals. So I will go back that topic later. So our study site is actually between Toledo and here. There's a huge wetlands called Ottawa National Wildlife Refuge. So surrounding area is a heavy agriculture, and then the wetlands start somewhere here, and then it eventually goes to Lake Erie. And then you can see that here, from all the way here, is very beautiful beaches. So site 1 is the beginning of the wetlands, and site 2 is in the middle. So site 1, this one is Dr. Chris Ray, who was my doctoral student. He did this research, his PhD dissertation. So site 1, and site 2 is in the middle. So the water gets quite slow, and then a lot of vegetations. And then site 3 is where the wetland meets Lake Erie. So we did a 1-year study, so we covered entire season. And then site 4 is actually the beach area, but sorry about the picture because we took in the winter. Chris first focused on nutrient and fecal contamination and decay reduction. So the summary of his finding is the total in site 1 is at the beginning, and then site 2 is the middle point, and then site 3 is where the wetland water enters into the lake, and site 4 is actual beach area. So this one, x-axis shows sampling date, 1-year date, and then this one is total phosphorus concentration. As you can see, the water travels from the entrance of the wetlands all the way to the beach area. Phosphorus level goes down. How about E. coli? E. coli indicates fecal contamination and potential presence of pathogen. So E. coli levels show the similar trend from site 1 to the site 4, showing decrease. So we have more data, but these are the highlights. So we validate that ecosystem surface benefits, not just for the environment, actually it's a human. And then also the reducing total phosphorus in E. coli from unsafe to safer level for human exposure during the summer swimming season. And then the next question is, wetland provides that nutrient reduction, but this one is heaven for a lot of wildlife, including migratory birds and geese. And then they do a lot of shedding, fecal shedding. So it gives, the question is, actually is a pathogen loading is happening in here, then what is the actually net effect? So wetlands can provide ecosystem service, but also a lot of animals. So if I'm a microbiologist, from my perspective, what is the actual net effect? So the public health concerns came from because all those, especially the birds, it's a really highly dense population of birds there. So there might be some emerging, re-emerging infectious agents coming actually load into the wetlands. So we need to find out, and then whether actually it really cancel out the benefit or still it's good. So that was the research question. Especially the water enters into the beach area where human direct exposure happens. So from public health perspective, that was a significant concern. So we need to check. So the question is, okay, wetlands and then pathogens. And then another reason behind it is we studied about pathogen loading from Canada geese feces in freshwater. And then we found that especially geese can carry a lot of pathogens. And then also another student, we studied about what geese have. So one of the primary concern was sugar toxin producing E. coli. Those are pathogenic E. coli. So then actually geese carry a lot of them. So if we are infected that, our immune system is not good, then it can really, this one is very, very much pathogenic. So then those we found is actually serotype that can cause disease outbreaks reported in the United States. So we have tons of rationale we need to look at. So then those, not just bacteria, but avian influenza and then other cryptosporidium, those are the ones also wildlife can carry who exist, who live in those wetlands. So one of the pathogen we kind of focus on is Arcobacter, which used to be Campylobacter. So those are emerging, but also the reason why we focused on this, those are zoonotic threat. Zoonotic means it's originate from animal, but those can cause infection and disease in human. So then geese is the one of the kind of vector to transmit those zoonotic pathogen. And another reason we looked at Arcobacter because Arcobacter was the culprit of outbreak in Ohio, South Best Island. And then the etiologic agent, one of them was Arcobacter. And then also Jason was involved in this study, but we did Lake Erie Beach study, and then we found that Arcobacter is so much prevalent in Ohio Beach, especially along the Lake Erie shore. And then we found that Arcobacter is related fecal contamination, and then also Lake Erie beaches are kind of potential reservoir of those pathogens, pathogenic Arcobacter. So we have also so significant of rationale. We need to look at Arcobacter as a kind of pathogenic kind of representative in this water. Then wetland can do something. So we traced the Arcobacter, but this time we kind of pulled the sites, upstream sites and downstream sites. And this one is Lake Erie Beach sites, and then this one is pre-wetland proxy sites. And then again, this is Chris, and then we did all the in situ measurements in the field. And then he brought back water samples to the lab, and then we did multiple analysis, including physical, chemical, and then also molecular microbiology measurements, and also we used ELISA. So just showing the summary, we focused on E. coli and then Arcobacter. So this one shows E. coli concentration, and the bottom one is Arcobacter. So the E. coli, the upstream site versus downstream, if we compare the median concentration, we saw that about 10 times decrease when shown in downstream sites. In terms of Arcobacter, then upstream and downstream, again, about 10 times decrease by the time the water reaches the beach sites. Okay, so I think the audience are more interested in this. So the wetlands and harmful cyanobacteria, because the nutrients are there and the water kind of stays there a little bit. And then nutrients from like a agriculture area, but also animals, like their waste can contain a lot of nutrients. Then what's going on in the water in terms of cyanobacteria? So this one is kind of the same study sites. Again, this one is 1 in the middle, and then site 3 and 4. So this one was mostly done by postdoc Chen-Nin Hu, who is an expert of cyanobacteria, especially toxin measurements. And then what he's holding is actually microcystis culture. So that's why microcystis shows that kind of blue-green color. So when microcystis is predominant in bloom, right, that's why we see blue-green color in the water. And then also that's why when we use phycocyanin, we see often cases very good correlation between phycocyanin and toxin concentration. But before I go this, let me kind of explain a little bit about microcystis. So when microcystis is one of the predominant species that make microcystin, but not 100% of microcystis are toxic. So don't be panicked if we find microcystis in our water, because some of them actually toxic, some of them are not. So that's why Chen's work was, what is the actual population dynamics within microcystis that generates toxic bloom? What we found was they also have genetic diversity like a human do. So from microcystis to make a toxic bloom, actually their genetic diversity of their toxin-producing gene pool is needed. So not just abundance, but also their genetic diversity of toxin-producing gene is very important. So toxic bloom is the product of all the combination and interaction of their abundance plus their genetic diversity. So that was the paper Chen published. And then when we studied that, we also generate this microcyst abundance data. So this one, site 1, we hardly detected microcystis. So this figure only shows site 2 and 3 and 4. So again, 4 is the beach water, and then 2 is in the middle, and then 3 is where the wetland water meets the lake. So site 2 is always show, not always, but usually showed highest level of microcystis among those 4 sites. So you can see that the x-axis shows the dates, and the y-axis shows the concentration of microcystis. So site 2 is the one we saw microcystis there, but when the water flows through the wetland, now by the time the water goes to the beach water, then it goes down. So of course, there is dilution factor, of course, yes. But that's what we see. So that's why we need to look at actually site 3, right? Site 4 is actually more like diluted by the lake water, but the site 3. So the microcystis abundance got reduced. But we are more interested in those toxic ones, because those are the ones that have the gene that make toxin, like in this case, microcystin. So when we look at toxic microcystis abundance, and then comparing the sites, again, this one is the date, and then this one is concentration, and then these are all different sites, and then 2 and 3 and 4. So looks like its general tendency is toxic ones got reduced too, but the kind of scale is a lot different. So the conclusions from our wetland studies, we looked at different aspects. First one is fecal indicator nutrients. Second one was other enteric pathogens that can cause diarrheal disease. And the third one is harmful algal bloom related factors. So I didn't present other chemical parameters, but also our chlorophyll data and phycocyanin data also show that kind of similar trend. So the conclusion was, among the wildlife, Canada geese appear to be play a limited role in contaminated water in the study area. So in the middle, actually, all the kind of their fecal poop related parameters went up, but by the time the water leaves the wetland, it kind of went back down. And then also cyanobacteria and toxin-producing cyanobacteria levels decrease by the time the water enter into the recreational area. So this one is kind of conclusion. So water cleansing ability of these wetlands will outweigh contamination arising from the area wildlife, in this case, Canadian geese, and the nearby agricultural lands. So these wetlands, all our national wildlife refugee wetlands, are providing important ecosystem service benefits for human and environmental health. So this one is the major kind of conclusion. So wetlands environment should be conserved or is re-established in close proximity to surface water that serves as drinking and recreational water sources. So I know that depending on what kinds of wetlands and then what kind of animals are around there and then what kind of water resource that wetland is used. So the all kind of site specific, but from our study, we came to this conclusion. So the initial concern, we know that ecosystem service that wetlands provide is good, but what if all the pathogen loadings and then they come and go? So whether still it is effective. So at least the bottom line is that at least it doesn't cause additional problem because of all the inhabiting wildlife. So that was our main conclusion. But also it looks like under that, at least during our study period, the wetlands was good for reducing cyanobacteria and toxic cyanobacteria. So the funding source is Lake Erie Protection Fund and the EPA Great Lakes Restoration Initiative funding and Ohio Sea Grant. And then I really appreciate the support from ONWR Ron Hoffman, also Scott Fletcher from ODNR. So these are the students who are involved in those studies. Thank you.
My name is Mark Crosby, Chippewa Lake. It's a small inland lake. We have cyanotoxin blooms. My question has to do with Department of Health regulations in the state. Department of Health regulations.
Mm-hmm.
EPA and ODNR mandate the posting of a sign when cyanotoxins are 20 parts per billion or above. However,
That's
across,
yes.
across the state, though, there is no mandatory closing of recreational waters at that level. And so my question is, given your research, is it possible that the Ohio Department of Health may start mandating closure of public beaches and recreational waters when toxins are above 20 parts per billion?
Okay, so the question is whether the state agency, especially ODH, has mandated the beach closure. I think that's kind of similar to what we do with the E. coli level, so the recreational water activity advisory. So I think for that, I can talk with ODH, but I think Ohio, the state, kind of follows. I mean, they can do. They can kind of establish their own guidelines. So that's why Ohio is the state that has the most stringent cyanotoxin guideline in the United States. So Ohio is the only state that has mycocysteine, cylindrospermopsin, and another cyanotoxins. But I'm not. I don't think I can answer that they can mandate or they should mandate, because U.S. EPA, they don't put cyanotoxin it is in the list, contamination kind of list, but it is not the list that can be actually regulated? I think that's a good question.
I have a question too. In your sites, from site 1 to site 4, as you go downstream, and you start to see decreases in concentrations of bacteria and toxins and things, as you go that direction, you also have quite a bit of dilution as
Yes.
you get to the lake.
Uh-huh.
Have you accounted for that at all? So is it, the last thing I want to do is downplay the benefits that wetlands have, but at the same time, I'm curious if that's anything you had looked into, is just the dilution factor.
I think that, so that's why we have not just the site 4, but 3 is the one. So 3 is the one actually, if you know the kind of all the hydrology, sometimes the lake water goes in and then back and forth. So we look at the net result, but so that's why we do like every weekly actually sampling and then see the kind of general trend. So I think definitely we cannot really ignore the dilution effect, and then also that, so truly it is a wetland thing or just a dilution effect. So actually we have another study using constructed wetland, actually Olentangy wetland, and then we did a similar study whether pathogen, we looked at all the parameters with Olentangy wetland, and then we, at least enteric pathogens, we didn't do cyanobacteria cyanotoxin with constructed wetland because Columbus, that wetland did not have the problem of cyanotoxin. But based on the E. coli data and other pathogens, so we saw the kind of, it doesn't really increase. So at least it stays by the time the water leaves the wetland, the concentration of enteric pathogens remained that range. So all the wildlife, they dump a lot of things, but actually it doesn't really kind of offset the impact.